Welcome to the first session of Genomics Data Analysis, where we are going to discuss how we can process, analyze, and interpret genomics data in general. So in today's session, we will focus on the code, DNA code itself, and we will focus on how to interpret the variation. That is the main focus of analyzing these genomics or these DNA code themselves, and what are the different ways in which these variations can tell us or can help us understand the biology behind the subjects or biology behind the study samples that we are investigating, that we are studying. So let's start by going through a couple of slides of review. So if you already know this, you can speed it up, or I'll follow it along. So let's start reviewing the fundamentals of genomics. Genomics is all about DNA, so we know that. Right? So let's also review what we know about DNA code and what are the answers that we can find inside the DNA code. And we also know that a DNA code is they comprises of huge sequences of nucleotides that are stored in this DNA. And how do we make sense of these huge number of sequences, and what do we do to make sense of them? What do we mean by making sense of this DNA code? Right. So if we attribute the DNA code or genomics data to a book, then the code can be thought as to be organized from letters that will form sequences, sentences, and the sentences will form paragraphs, and paragraphs are organized into chapters. And understanding and knowing how to read this code can help us decipher the full story about who we are, can help us understand to look into our history, can also help us understand how we as an organism or as a being are connected to other living things that are around us. So when we talk about genomics, the first thing that comes to our mind is genes or codes. These codes or genes are the ones that are passed from cell to cell, and we have approximately 3.2 billion letters in total, and these encodes to around 22,000 proteins. And these genes are made up of biochemical entity called DNA, which is a short form for deoxyribonucleic acid. This DNA molecule is a double helix, that is two long thin strands that are twisted around each other like a spiral staircase. So in this DNA, the backbone of each strand is made up of sugar and phosphate, and these two strands will interact with each other through nucleotide bases. That is what we sequence. So as we spoke about that, this DNA is made up of nucleotides. So the nucleotides themselves are made up of three important components: a sugar ring that is called deoxyribose, a nitrogen containing base, and a phosphate group. These nucleotides are the ones that are joined to one another in a chain by covalent bonds that form between the sugar molecule of one nucleotide and the phosphate group of the next. This results in an alternate sugar phosphate backbone, and the nucleotides of the two complementary strands are connected together. And this connection that connects two nucleotides of the complementary strand is called base pairing or base pairs. The base pairing connects these two strands of DNA, and this connection gives to the characteristics double helix form of the DNA. So in this example, in this structure, we can see that the adenine is always bound to thymine, and the guanine is always bound to cytosine. And they form the four bases of the nucleotide A, T, G, and C. So that's a very short introduction about the DNA, and its basics. So let's look into various ways where we can spot the DNA variation, and how can those be helpful for us in understanding the biology behind it. So we spoke about this, and what do we mean by studying the DNA variation? When we discuss about studying DNA variation, we always want to compare the sequences that we have in the current sample that we are that to the reference sequence, and to identify what are the places in which the sequences or the bases vary or bases differ generally. So that this differences in the bases or this differences in the nucleotides or this differences in the in this DNA code can give rise to unwanted effects, which we call as mutations, or typically they are termed as DNA variants. Right? When these variations affect the end product, which could be a protein coded by the specific gene, then we will call this variance as mutation. We will speak about this a little bit later. And depending upon what type of variant that we are getting, the end product, the protein can be altered in their sequence, and that alteration can affect the functionality of the protein, and hence we are ending up with a pathogenic variant that is termed as mutation. So the reference sequence, where do they come from? So this reference sequence actually comes from the first human genome projects, which is what enabled us to compare any given DNA sequence from a human to a common reference. This common reference is what is compiled from multiple individuals, and this project is a 13-year-long project which started at 1990, and we all know about what this project gave us till date. So the primary goals of this human genome project was first to determine the DNA sequence and the location of the estimated genes. The estimated number of genes at that time was around 100,000. But now we have filtered that, and we have come to a consensus that there are around 21,000 to 22,000 genes in our whole genome, and all the other sequences are present to regulate the translation of these genes into proteins. Most of the other sequences. So this is the reference sequence to which we will compare any new sequence that we will generate, and identify the differences in the sequences and try to characterize the differences in the sequences, which in a nutshell can be termed as genomics data analysis. So this resulted, or with the advancement in the next generation sequencing technology, we have now actually obtained, or there is an ever increasing number of genomes that are sequenced, and the cost of the sequences have drastically decreased, and this gives rise to more number of sequences being generated, and we in the end are ending up with more and more data that has been deposited to public repositories like NCBI and EBI, and we can make use of all these public repositories to make meaningful data analysis and understand the genomic variations that are present in the samples in general. So this plot indicates that the decrease in the cost of genomics technology generally followed Moore's law until one point, and after that it drastically reduces, and which can actually give you, or which can actually give you an indication of how the technology has genomics technology has improved leaps and bounds. So what do I mean by understanding the differences? We already spoke about that. A little bit more background for this. Efforts such as the one described in the previous slide, variety of different, numerous different studies that will try to understand variances or variations that in the genomics code compared to the reference code, it can now, for example, detect harmless and also harmful variances present in the sequences that we are actually sequencing. Just like any code, or just like any sequence of letters, we can expect the DNA to contain mistakes, and these mistakes actually occur as a result of copying the DNA source too many times, and also these mistakes can also occur as a result of various stresses that the cells will undergo, and these stresses and the result of copying may actually interfere with the copying mechanism of the DNA and can introduce mistakes. And these mistakes are the one that we are focused on detecting. And when these mistakes actually occur in important regions like genes that will encode very important proteins, and even inside the genes, important stretches of sequences that are that result in key changes in the proteins that will end up altering the shape and size of the protein, and it can also end up altering the shape and size of the cell as it is given here, can actually lead to serious issues when that gene is being produced. This is expertly understood in the event of sickle cell anemia, where a normal protein sequence encoded by the gene actually produces red blood cells of a normal morphology, and when one single nucleotide change actually changes the amino acid from glutamate to valine, and that results in producing red blood cells that shape like sickles and result in a reduced carrying capability of oxygen, and hence it results in anemia. So this is one of the example of mutation or point mutation, one of the examples of mutation that occurs in single nucleotide position. And all the mutations that occurs in single nucleotide positions are actually defined as point mutations. Depending upon what is being changed, there are several types of point mutations that one can characterize. So one among those is substitution mutation. A substitution mutation can cause several consequences to the DNA code, but this substitution mutation implies minimal change that only leads to single nucleotide change, as we actually discussed. So what is changed? One nucleotide that is in the reference sequence is substituted by another nucleotide, and this resulting substitution mutation may or may not end up changing the amino acid sequence that the trinucleotide codon codes for. And there are also other forms of single or point mutation, which could actually involve insertion of a single nucleotide into a position in the altered sample sequence compared to the reference sequence. This insertion, unlike the substitution mutation, may end up changing the reading frame, and that encodes a trinucleotide codon, and hence from the point where the insertion is observed, the resulting protein sequence may be completely different from the one that is present in the wild type or the reference genome. And this may result in the protein that may not function properly and or that may prematurely end. And contrary or complementary to the insertion mutation, we also have deletion mutation, where one nucleotide from the sample sequence compared to the reference sequence is deleted. Just as in insertion mutation, this deletion mutation will actually change the reading frame of the trinucleotide codon, which produces amino acid sequences, and hence it results in the protein sequences that is completely different from the wild type variant. And this may also, or this is also most likely expected to change the functionality of the protein and cause serious defects in the cell's functioning and general cellular molecular function. So unlike substitution mutation, insertion or deletion may actually contain more than one number of mutation, one number of nucleotide, and again in those instances the resulting protein may completely differ. And there is another form of variation or another form of mutation, that is duplication, which consists of a piece of DNA that is abnormally copied more than one or more times in different places or in adjacent location. This type of mutation again may alter the functionality of the resulting protein, and some of the examples of these mutation we will discuss in detail later. So depending, we spoke about point mutation, and focusing on the types of mutation from the point of view of the amino acid sequence that these mutation encodes for, we can again categorize these mutation into missense, nonsense, and frame shift mutation. So a missense mutation is characterized by the change of point mutation at the nucleotide level that result in the changes in the amino acid sequences, right? Sometimes this single amino acid change that occurs in the protein may occur at a structural or functionally important location, and may end up in greatly changing the protein's function. So in that event, as we spoke about, as we discussed, the end result may actually completely change the function of the protein and hence affect the proper functioning of the cell completely different. So a nonsense mutation is actually a change in the nucleotide base, prematurely stops the protein production by introducing a stop codon at that specific position. So this definitely changes not only the protein sequence themselves, but it stops the protein sequence production prematurely and results in a shortened length of the proteins themselves. So this can result in the protein folding incorrectly or not folding at all, and hence the functionality of the protein is completely lost. A frame shift mutation is what results in when we have an insertion or a deletion, as we already discussed, and this changes the frame which reads the trinucleotide codon that encodes for different amino acid sequences. And hence, since the reading frame is changed by the insertion of this one adenine at this position, now whatever is encoded after that insertion position or after that specific point mutation is completely changed, and this again will result in a protein sequence that is different from the wild type, and this differed or this mutated protein sequence may again result in incorrect folding or result in a completely different function than what the original protein is intended for. All these will actually affect the cell's functionality to a larger extent, and depending upon what is where this changes occur in the morphology of the tissues and how they actually occur and how they actually affect the cell and tissues and hence the organism, they can be characterized into genetic variation compared to mutation. So generally, all the changes that we observe in our sample compared to the reference sequences are termed as genetic variations or genetic variants. So the genomics data analysis focuses on identifying these list of genetic variations and characterizing, annotating them into potentially harmful and potentially not very harmful, and that the one that can be ignored by just characterizing them to a different level. So for example, if you see the morphology of this, for example, in this example, we have a wild type form, which in its natural form, without any mutation, has this specific morphology. And there could be some variations in their genetic gene sequences that can change, for example, the eye color, that can give a couple of more modifications to the morphology. But if these mutations are, no, not mutations, if these variations are not going to change the functionality or not going to affect the survivability of that specific form or organism, they are not termed as mutation. They are generally considered as genetic variants or polymorphism, etc. But variants that results in loss of function, that results in improper function, that can cause diseases or that can cause genetic defects are termed as pathogenic, and hence they are called as pathogenic mutations. Sometimes in several of the publications, the word mutation and variants are actually used interchangeably, but generally when you refer to a mutation, we always refer to the pathogenic version of the variant. And when you refer to variants, we either refer to changes in the nucleotide sequences that are not characterized as of now or as of at the moment of data analysis, or they are characterized to be inconsequential to the functionality of the tissue or organism. So that is the difference in their terminology that we have to keep our mind to. So as we spoke about, how do we identify these genomic variations, right? These genomic variations can be identified by subjecting the samples into high throughput sequencing or next generation sequencing, which you will learn a lot in the upcoming sessions. And when we map all these reads to specific position in the genome or reference genome, then we can identify the places or the positions in which that specific nucleotide is changed, or that specific nucleotide is converted into a variant or a mutant form. When we speak about single nucleotide variant, so when we speak about the genomic variation that we are trying to characterize, let's refresh our memory that what we have been discussing and summarize into 4 or 5 points. So single nucleotide variant, or SNV in short, can be characterized as a single nucleotide change that may or may not be present in whole or most of the population. Basically, this means any change that we observe in our sample from the reference genome can be termed as SNV, or single nucleotide variation. While another term that we always hear or that we always discuss is single nucleotide polymorphism. For example, when we have samples that typically consist or comprises of more than 1% of the population, or more than, for example, not more than 1% of the population, and when we observe this specific variant in more than 1% of the population, and that variant is considered to be single nucleotide polymorphism. So when we refer to this 1%, that refers to the whole population set of that specific group of sample that it refers to, not the sample themselves. So now you can, for example, appreciate the amount of sequencing that has to go into estimating the 1% change of variant that is present in 1% of the whole of the population, to term them as SNP. For example, the SNPs that are characterized for the ethnic group and different races like Caucasians, different races like Asians and Europeans, they actually involved in sequencing more than 1%, or approximating to sequencing more than 1% of the population, to identify these single nucleotide polymorphisms for that specific group of samples or population. So any variant that we find to occur above the level of technical error can be termed as single nucleotide variant, and any single nucleotide variant that is prevalent in approximately 1% of the population can be termed as single nucleotide polymorphism. And another type of variation that we often refer to is the copy number variation, where a gene is encoded in multiple copies in our genome, but the number of the copies varies between different samples and between different sequences. And when this number of copies actually varies significantly between the reference sequences and our sample sequences, then that may also result in the changed level of gene expression, and hence may actually result in altered phenotypical condition of the cell or tissue or organism in general. So all these changes that are characterized at the level of the genes or genome are termed to as genotype, and many of these genotype can actually end up in specific phenotype that can be harmful, or that can be harmless, or that can be potentially ignorant, or potentially beneficial in some aspect. Right? For example, in this specific gene, one of the examples of harmful mutation or harmful genotype is the one that involves PAX genes. In this specific example, PAX6 is a gene that will encode for PAX6 protein, and it has been characterized and recognized that this PAX family of genes is necessary for the development of eye in almost all of the organisms. And surprisingly, the development of eye in many of the organisms follow similar developmental path. So we can follow the progression of this gene and their defects in many different model organisms. And experiments in such organisms like mouse or mice samples demonstrate that deficiency in PAX6 will lead to not only in decrease in the brain structure of normalcy or normality that leads to autism. It can also lack complete formation of iris, or it can result in cornea, and that will end up causing blindness in the blindness in the variant or in the phenotype, the blindness as part of the phenotype. This is one of the examples of harmful mutation. There are a couple of examples of potentially harmless variants. For example, some mutation in specific gene can cause long hair growth in eyebrows or in eyelashes. They can be potentially a little bit difficult to deal with, but they are not harmful in any case. One more example of, for example, potentially not very harmful, but advantageous mutation variant is the SNP that is linked to or linked with the development of blue eyes for the Caucasian is the variant that is linked to the blue eye of Caucasian is actually an SNP that is characterized for European and US race people. So these are all different ways and different forms of variants that can or not that may or may not end up in causing difference in the phenotypical versions of the samples. So such analysis to understand and characterize these variants can be performed at many different levels to investigate the functions of variety of different genes in multiple different research areas. Right? These research areas can include drug metabolism by studying the enzyme function that is key in the catalysis and the key in the functionality of specific metabolite. And this can also end up trying to characterize, we can also end up trying to characterize mutations in specific genes that can cause tumor. In that case, genes that are linked with tumor progression and tumorigenesis. And we already spoke about sickle cell anemia. This in case, this in this example is caused by the effects of germline mutations that are inherited from our parents. So furthermore, one can also use genomics to explore pathogens and to understand important properties of their pathogens as a result of their, or as a function of their genomic variants, to study virulence, to study disease transmission, and to study drug resistance, etc., etc.